afternoon. Good morning, everyone. Thank you very much for joining us. Welcome to this HDSA research webinar where we're going to hear an update about the PIVOT HD trial being conducted by PTC Therapeutics. My name is Leora Fox. I am the Assistant Director of Research and Patient Engagement at HDSA. I am joined also by our Chief Mission Officer, Dr. Eric Johnson, as well as our speaker, who I'll introduce in a moment. But first, I'm going to walk through a couple of slides to just remind you how we approach our Q&A and let you know how to find the recording afterwards and learn about some other resources and upcoming webinars. So we'll wait until after Dr. Klein, our presenter, has spoken to answer questions, but at any time during the presentation, you can type your questions into the chat box in your Zoom toolbar. And this is a closed chat, so only HDSA and our panelists will be able to see what you've asked. So we'll pose the questions and we'll address as many questions as we can and do our best to follow up afterward if needed. We are recording today, so if you want to revisit this presentation or send it to somebody else, you can go to hdsa.org/researchwebinars, or you can go to HDSA's YouTube channel using the button at the top of our homepage. Usually it's up, if not in a few hours, then within a day or two. We have an upcoming disability chat webinar on Tuesday with Allison Bartlett, and that will focus on disability options and resources for young people with juvenile HD. You can go to hdsa.org/disability to learn more and register. As a reminder, we put the word out about upcoming webinars as well as our weekly research blog and many other things on all our social media channels. So please do follow HDSA there for updates. I'd also encourage you to check out HDSA's hdtrialfinder.org to learn more about currently recruiting clinical trials. And right now on our surveys page, there are many opportunities for anybody from an HD family to participate in research from home. Some of these have different kinds of incentives. These researchers really need your voices. So you can do that at hdsa.org/surveys if you want to participate and lend your voice. So now I'm very pleased to introduce to you our speaker from PTC Therapeutics, Dr. Matthew Klein. He is PTC's Chief Operating Officer. He is an experienced leader in biotech and previously in academic medical research. And today he's going to share with us the latest updates from the PIVOT HD trial, which is testing an oral genetic therapy for HD. So I'll hand it over to Dr. Klein.
Thank you very much, Leora. It's a pleasure to be here today, and I'm incredibly grateful to have this opportunity to share this update with the HD community. I'd like to begin with an introduction to PTC Therapeutics for those who don't know us. PTC was founded 25 years ago with the single purpose of using innovative science to develop therapies for patients with rare diseases. We've grown quite a bit over the past 25 years. We now have a presence in over 50 countries around the world, and we have over 1,300 employees. But one thing hasn't changed. We remain committed to using pioneering science to develop drugs for people who desperately need them. That was our focus when we started, and it remains our focus today. We have a broad portfolio of therapies that are at different stages of development and several that are on the market now. Our drugs take different approaches to treating diseases, and also we treat a number of different diseases, including Duchenne muscular dystrophy, which is the first disease we focused on when the company started. We have a gene therapy program with a recent approval of the first ever gene therapy that's directly administered to the brain. We have drugs that target metabolic diseases as well as cancer. But the focus today is on our splicing platform, which is really quite an innovative approach to treating diseases. In our splicing platform, we use oral medications to alter the genetics in patients to help treat their diseases. And the first splicing therapy we developed is Evrysdi for a disease known as spinal muscular atrophy. And I'll be talking a little bit about that program shortly, and then I'll go into a deeper discussion about our next splicing program, PTC 518, for the treatment of Huntington's disease. So Evrysdi, as I mentioned, was the first compound from our splicing platform, and it's also the first ever splicing molecule that has been successfully developed. It's currently approved in over 90 countries, and it's approved for the treatment of a genetic disorder known as spinal muscular atrophy. And it's approved for types 1, 2, and 3 of spinal muscular atrophy. And currently in the United States, it's approved to treat patients as young as 0 to 2 months of age, which means it's able to treat patients with or babies with spinal muscular atrophy before they're even symptomatic. And it's currently the most widely prescribed therapy in the US. I bring this up because the development of Evrysdi was based on years and years of science and pioneering in our laboratories at PTC. And it's through that extensive experience we have in the science of developing splicing molecules that allowed us to bring Evrysdi forward and to show that with that really good science background and scientific efforts, we can develop a therapy that can get approved in the United States and around the world and treat a very serious disorder, spinal muscular atrophy. I just want to take a minute to explain how Evrysdi works because it's actually incredibly interesting. So let me first start by talking about what happens in spinal muscular atrophy. Babies born with spinal muscular atrophy have an inability to produce a protein known as SMN1. Without the ability to produce SMN1, there is an inability to have muscle function throughout the body. That disease can present early in life or later in life, but regardless, it results from an inability to make a very important protein known as SMN1. What we were able to do with Evrysdi was basically take a protein that people with SMA have called SMN2, and with a pill, force the production from SMN2 to SMN1. So basically, an extra piece of protein is put into SMN2, and as the result, it now turns into SMN1. So basically, what we are able to do with Evrysdi is take advantage of something known as splicing, where we can alter production from a protein the body has and may not need as much of to form a protein the body doesn't have and really, really needs. And we've done that to basically be able to treat the disease of a deficiency in the protein of SMN1 known as SMA. We learned a number of very important lessons from the many years of development of Evrysdi. We learned a great deal about how you can take a splicing molecule, make sure it has several important elements so that it works well, and importantly, that it's safe. And then how do we bring that molecule through all the steps of drug development before entering the clinic? And then once it's in the clinic, how do we set the stage so that molecule can be successfully developed and be available for patients? And this included elements of ensuring that when we develop a drug for a disease, that it's very specific for that disease, so that it's only targeting the genes that are impacted by a specific disorder. In the case of Evrysdi, it was spinal muscular atrophy. We also learned how do you make sure that these molecules get to the right places in the body so they could do their job. So in diseases like SMA, where you have, and of course, Huntington's disease that we'll talk about, where you have the brain and the central nervous system affected, it's very important that we make these molecules in a way that enough of the drug gets to those important places so that we can effectively treat the disease. And this, in the case of, again, both SMA and Huntington's disease, that's the central nervous system. We also needed to understand how best we can set up a program that once this drug gets into the clinic, that we can study it and move it as quickly as possible through what can be a very long drug development process, so we can prove that the drug is safe and we can prove that the drug is effective, so we can get it to as many people as possible affected by a particular disease. So coming on the heels of the successful development of Evrysdi, we asked a very simple question. Can we leverage those learnings from Evrysdi's success to develop a drug for people with Huntington's disease? It became very clear to our scientists early on, many years ago, that some of the features that allow us to develop a splicing molecule for SMA could also allow us to develop a splicing molecule for Huntington's disease. As I'm sure you're all aware, Huntington's disease is a monogenic disorder, which is caused by a mutant protein, a mutant Huntington protein. And the question was, can we use splicing to not increase a protein like we did with SMA, but do the opposite, decrease a protein decrease the mutant Huntington protein that is causing Huntington's disease. And so the idea was, can we use splicing to simply make less mutant Huntington protein and slow disease progression in Huntington's disease? So let me show you now how that mechanism works for PTC 518 in the treatment of Huntington's disease. So first, looking at this part of the slide, we see what happens in the genes in the disease state, which is we have a CAG expansion in exon 1. And basically, this protein gets produced that is a toxic mutant Huntington protein. It turns out in the gene for Huntington's, there's this sequence called a stop exon that doesn't get expressed. Basically, as this mutant protein is produced, the only thing that gets produced is the parts of that gene that make the mutant Huntington protein. So for us, we ask the question, can we do something to take advantage of the fact that there is a little stop sign here to basically make that stop sign work and stop the production of the mutant Huntington protein? And so this is what happens when we have a splicing molecule that targets the Huntington gene. We basically use a pill, in this case it's PTC 518, to put that stop sign right here. And by putting that stop sign right there, we're able to stop the production of the mutant Huntington protein. So just to repeat that, what we're doing is we're taking advantage of the fact that there is an unexpressed stop sign in the Huntington gene. And we're using, in this case, PTC 518 to introduce that stop sign to stop the production of the mutant Huntington protein and essentially reduce the levels of the mutant Huntington protein and hopefully ultimately impact Huntington's disease progression. As I mentioned, we learned a number of very important lessons from the development of Evrysdi that we've put in place in the development of PTC 518. As I mentioned, this is an oral therapy, but it's very, very important that if we're taking an oral therapy, that it gets to the brain, gets all around the brain, and gets to the brain in sufficient quantity so that it can have an impact on the disease. And so this was something that our scientists spent a lot of time on, building a drug, constructing a drug that had the right structure so that we were certain that it can get to the brain and get to all over the brain and to high enough levels that it can have an impact on Huntington disease production and impact disease progression. This included making sure that it got to, as I mentioned, all regions of the brain in equal amount, because we know that Huntington's disease is a total brain disease. And if we're going to lower Huntington protein and have an effect on disease progression, we're going to need to make sure that we're lowering Huntington protein all over the brain. We also need to make sure that it's not effluxed. What does that mean? Well, when drugs get into the brain has a special security system or defense system that it likes to kick foreign things out. So we had to make sure that when PTC 518 gets into the brain, that it stays there. It doesn't get kicked out. So it stays there long enough, again, so that it can have an effect on the production of Huntington protein throughout the brain. And finally, very important, we wanted to make sure that PTC 518 was highly selective and specific for the Huntington gene. This is important to ensure that it's getting to the right gene to have an effect, but also not affecting other genes. We want to make sure that it's only really working on the Huntington protein, so that selectivity and the specificity for the Huntington's gene is very important to having a therapy that's not only effective, but is also safe. We did a lot of work before bringing PTC 518 into the clinic to ensure that we had developed a molecule that does exactly what we wanted it to do. So when you look first at the top part of this slide, you see that we wanted to make sure that we're getting a molecule that gets into the brain and can do exactly what we hope it ultimately is going to do for people with Huntington's disease. That is, reduce the amount of mutant Huntington protein that's produced. And so what you see on the top is that we have what's known as a dose-dependent reduction in Huntington protein. Very, very important. That tells us, this piece of data tells us that we have a molecule that is getting into the brain. In this case, it's of a BAC-HD mouse. It's getting into the brain of the mouse, and it's doing exactly what we want it to do, interacting with the Huntington gene, putting that stop sign in place and decreasing the production of the mutant Huntington protein. And seeing this dose-dependent reduction is something that we as drug developers really look for, to know that we have something that's acting very specifically and reliably, and that we can then adjust the dose to get the right level of lowering. On the bottom of this slide, you see another important element of the molecule that we wanted to make sure that we achieved. That is getting uniform lowering throughout the brain. As I mentioned earlier, we know very well that Huntington's disease is a total brain disease. And in order to have an effective therapy, it's going to be important that we're lowering Huntington protein, the mutant Huntington protein production in all of these different regions. Because you can imagine, if you're only lowering mutant Huntington protein in one part of the brain, you're still making the full amount in all the other regions. And obviously that could compromise the effectiveness of any therapy. So what we were able to capture here is that we were developing a molecule that was not only resulting in a dose-dependent lowering of Huntington protein, but it was lowering Huntington protein levels consistently in every region of the brain. So with these basic science data and the ability to confirm that we had designed a molecule that was selective and specific for the Huntington protein, was getting into the brain in high enough levels to achieve our desired effect, and that we were getting the broad distribution throughout all the regions of the brain to allow for the uniform lowering of Huntington protein, we then move forward into the clinic. And the first studies that you do when you're in the clinic are known as phase 1 studies, single ascending dose or SAD, and multiple ascending dose. And these are usually done in healthy volunteers. And so we initiated the phase 1 program almost 2 years ago, and we did both the single and multiple ascending dose studies. We wanted to check for safety, make sure that the medication is tolerated, and also see what we're getting in terms of levels of the drug in the blood as well as the brain, so we could start understanding what dose levels we might need to then use in future clinical trials to treat patients. We were very happy to report that in both the single ascending dose studies, that's when you just give one single dose of the drug at different dose levels. So in this case, we gave to healthy volunteers one pill at 5 milligrams, 15 milligrams, 45 milligrams, all the way up to 135 milligrams. And we saw that we were getting a dose-dependent lowering of Huntington RNA. And then we did the multiple ascending dose, which is basically giving 14 days worth of doses at different dose levels. And what we saw was a dose-dependent lowering of the Huntington protein levels. As I mentioned earlier, seeing this dose-dependent lowering is a very important check for us, that we have something that's not only getting to the gene, but that it's actually having the desired effect, which is lowering the Huntington protein production. We also wanted to make sure that the drug was getting into the brain. Again, I mentioned several times this importance of ensuring that enough of the drug gets into the brain so that we could have the desired effect where it matters most to the disease. And in the case of Huntington's disease, that's the brain. We had observed previously in animal studies that we were getting basically a 1-to-1 ratio of drug levels in the blood to drug levels in the brain. That's exactly what we'd want to see to make sure we're getting enough of the drug into the brain. What we saw in healthy volunteers, though, was that we were doing even better. We were actually getting higher levels of the drug into the brain than in the blood. And that is really important for us, because not only does that verify that we have a drug that's getting into the brain and staying in, but also that we could potentially use lower doses in patients to achieve higher levels of Huntington protein reduction in the brain. So in summary, phase 1 was very successful for us in that we showed that the doses were safe and well tolerated. We were able to get predictable drug levels in the blood. We saw the dose-dependent lowering of the Huntington mRNA and also a protein, and that we were seeing that we were getting very nice levels of drug into the brain, consistent with what we believe we need to have in order to have a potential benefit to patients. So with phase 1 completed, we then moved into the phase 2 Pivot HD study, with which I think many of you are familiar. The objectives of the Pivot HD phase 2 study is to now demonstrate the safety, tolerability, and ability of PTC 518 to reduce Huntington RNA and protein levels now in people with Huntington's disease. Phase 1 was in healthy volunteers, and now we're moving into people with Huntington's disease. And we also want to begin to understand what's the effect of lowering Huntington protein throughout the brain on a number of important biomarkers of Huntington's disease. Again, all information that's very important to the successful development of PTC 518. The study design of the Pivot HD study of PTC 518 is as follows. We're doing a 12-month placebo-controlled study, and we're starting with 2 different dose levels, 5 milligrams and 10 milligrams, and we're also going to have a placebo group. The 5 milligram and 10 milligram dose levels are based on all of that data that we got out of the healthy volunteer study, including how much Huntington mRNA and protein reduction we had at certain levels, as well as understanding the relationship of levels of drug in the blood to levels of drug in the brain. We believe that starting with 5 milligrams and 10 milligrams puts us in a very good position to potentially be at a level that can provide clinical benefit for people with Huntington's disease. The first part of the 12-month study is 12 weeks long and focuses on understanding the drug levels and the response in terms of lowering of Huntington mRNA and protein in the blood, as well as confirming the ratio of drug levels in the blood to drug levels in the brain that we saw in healthy volunteers. The second 9 months of the 12-month study is focused on the biomarker effect of PTC 518, and this will provide us very important information in understanding what's the impact of lowering mutant Huntington protein levels in the brain on markers of disease we know that are important to understanding ultimately the effect on disease progression. We spent a lot of time in designing the study to make sure that we are enrolling a patient population that we think puts us in the best position to have a successful study. I want to make very clear that we believe that PTC 518 could be beneficial to people with Huntington's disease at all different stages of disease. The ultimate goal would be to have a successful development of this drug for all people with Huntington's disease, regardless of the stage of disease. But in doing a clinical trial, it's often important that we select a specific population of patients in whom we have the best chance of having success in this study. And so we spent a lot of time working with experts in Huntington's disease to try to identify the characteristics of an ideal population for this clinical trial. Again, I want to emphasize that the ultimate goal would be to have a drug for all patients with Huntington's disease. And so the inclusion criteria include age 25 and older, CAG repeats of 42 to 50, having a ISS score independence scale of 100, a normal TFC of 13, and then a PIN score between 0.18 and 4.93, which is basically a combination of a number of different tests to make, and again, this was really an important part of the inclusion criteria that we think brings us a group of patients in whom we can best capture benefit in the short 12-month clinical study. The primary endpoints are as follows: the safety and tolerability of PTC 518, as well as the % reduction in Huntington mRNA and protein levels in the blood. And these are information we'll gather from the first 12 weeks of the study. And then as we move into the second 9 months of the study, we'll focus on biomarkers of disease like CSF protein levels of mutant Huntington protein, changes in neurofilament light chain, as well as changes in brain volume on MRI, as well as a number of different clinical measurements that we'll be testing. That'll be very important for us to learn a lot about the potential effects of PTC 518 and also help us design the next clinical studies that we'll have to do for drug development. So I'm going to end by providing a quick update on the Pivot HD trial. We're actively enrolling at multiple sites in Europe and Australia. I also want to confirm that there have been no report of drug-related adverse events reported to date anywhere in the world. That's very good. That's showing so far that we are having a good sign of safety based on what's been reported to date. We've also shared that enrollment is currently paused in the United States, and I know a number of you are going to have some questions about that we can help answer. And I want to assure everyone that we continue to work with global authorities worldwide so that we can ultimately allow patients around the world to participate in the PIVOT HD trial. When we do clinical trials at PTC, we usually do trials globally. That is, we include many countries around the world. We also know that when you design clinical trials, that regulatory authorities in different countries often ask different things and have different requirements, and the timelines in some countries are sometimes longer than in others. And we're very fortunate to be in a position that we are in today, in that we have a trial that's up and running in a number of countries around the world, in a number of different expert centers in Huntington's disease, even though we're not quite up and running right now in the United States. So with that, I'll conclude, and I'm happy to take any questions that folks may have. And again, I just want to thank you, Leora and HDSA, for this opportunity, and also to share my gratitude to be able to partner with you as well as the Huntington disease community as we work to bring PTC 518 forward for patients with Huntington's disease.
Thank you so much, Dr. Klein. And I know that we have limited time today, but I hope that you will be able to stick around for at least a few questions, because there are some really great ones coming into the chat. And the first one I want to address is related to the US pause. As you mentioned, there's been a lot of questions from the community since that news came out recently. And so I wonder if you can speak in a little bit more depth about that, and what the FDA, for example, has asked for, how this affects the timeline. Someone has asked directly whether there is a timeline for this to begin again in the US with those 5 and 10 milligram cohorts, as you mentioned.
Yeah, absolutely. So maybe I'll just give an overview of the timeline of starting the study and how we arrived at where we are today. As I mentioned, we intended from the beginning, as we do in most of our trials at PTC, for this to be a global clinical trial, that we knew there's an interest in participating in clinical trials, and specifically an interest in PTC 518 worldwide. And what we did first is, when we were ready to start the trial, we had enough what's known as toxicology data. That's data that you get in animals prior to going into humans that helps give some idea about safety and doses that should be used in humans. And what typically happens is, if you want to do a 3-month study, you need to have done 3 months worth of studies in animals. If you want to do a 9-month study, you need to have 9 months worth of studies in animals. And actually, once you get beyond 9 months, usually 9 months is good enough. So if you want to do a 12-month study, typically you need to do the 9-month animal studies to allow a 9-month or longer, in this case, a 12-month study. When we were ready to start the study, we had the 3 months data that allowed us to start the 12-week portion of the study. That was the plan. And the plan was we would start the 12-week study around the world. We knew we'd be getting the results of our 9-month animal studies. And again, those are the animal studies that would allow us to do the full 12-month PIVOT HD study that we wanted to do. We got the data from those studies. We looked at the results, and we were very encouraged. We had what we believe were very good levels of between the doses and what the safe margins were in animals. And so therefore we move forward with providing the protocol to regulatory authorities for 12 months, the full 12-month study. I'm happy to report that this was cleared by regulatory authorities around the world: UK, Germany, Netherlands, Australia, many different countries around the world. However, when the FDA looked at the animal studies, they said to us, we'd like to see some more data before we let you move forward with the study you want to do, at those doses that you want to do, for the length of the study that you want to do. And so that put us in a position that we said, okay, we have to pause on doing any enrollment in the United States, because we want to do the full 12-month study in the United States as we're doing around the world. Let's keep the study moving outside the United States, because the most important thing we can do is get the PIVOT HD study enrolled and completed, so we have very good data to know if we're ready to move forward in getting PTC 518 further through the development path, and hopefully ultimately closer to approval. So we're in a position now where we are working with the FDA to see what we can provide them in terms of the data to help open things up again in the United States. Again, our number one priority, of course, is getting the study enrolled and done. And so we're really focusing a lot of efforts on getting the study enrolled at the sites we can right now while we work with the FDA, because we really want to obviously be able to give people with Huntington's disease in the United States the opportunity to participate in the trial if they're eligible and want to participate. So we're going to continue that work to allow that to happen. I can't give you a timeline right now to say exactly when that's going to be. I can tell you that we are committed to getting the study enrolled, getting the PTC 518 program moved forward, because we believe that there's a lot of potential for PTC 518. It's an oral molecule, gets to all areas of the brain. And I think we all believe that by lowering mutant Huntington protein levels, we can really have an impact on the disease. So we want to move as quickly as possible to get PTC 518 moving forward, get the necessary clinical trials done. And so if we can show that it's safe and show that it's effective, we can make it available to his patients with Huntington's disease around the world. So that's our number one commitment. However, of course, we want to bring the opportunity to the United States for patients to participate in the clinical trials. So we're going to continue to work on that as well.
Thanks very much for clarifying all that. That was very helpful. I want to also make sure to address several questions that are related to the comparison, really, I think, between PTC 518 and another oral Huntington lowering drug that is in development called branaplam. We learned in August that branaplam was unfortunately causing some neurological side effects in people who are involved in the Vibrant HD study. And so I'm wondering if you can touch on, there are folks who have asked things as specific as, is it the same pseudo exon, which I think is, really on the scientific side, to sort of, is the FDA concerned because of the results from branaplam? So maybe you can just touch a little bit on that.
Yeah, absolutely. So obviously branaplam has a similarity in that it is a splicing molecule, but obviously there's a number of really, really important differences. First and foremost, unlike, Pivot HD is up and running. It's not Vibrant HD. It's not stopped. The trial's up and running, and it's absolutely moving forward. In terms of other really important differences, I spent a little bit of time talking about how we learned from Evrysdi the importance that a molecule is selective and specific, that it really is specific for Huntington's disease. Now, it turns out that branaplam started in development for SMA, the same disease that Evrysdi was approved for. And then there were some issues with neuropathy in some of the studies with branaplam in SMA. And then there was a question of, could it be used for Huntington's disease? And so I think that tells you right away that it can't be that too selective and specific if it can be used for two different diseases, right? We had Evrysdi that was developed for SMA, 518 Huntington's disease only. So right away you have an issue with not having a molecule that may be as selective and as specific as PTC 518. And we know that's really, really important for having a safe and effective splicing molecule. I think there's also some very important differences about the ability of the molecules to get into the brain and stay into the brain. As I mentioned, one of the key design elements for PTC 518 was ensuring that it can get in the brain and doesn't get what's called as efflux, kicked out of the brain. And so we can say for sure that we have a molecule, obviously the data show that gets in and gets into the brain at very, very good levels, actually even better than we had designed it to do, which is really important. And I think that ability to get in and stay in the brain with that 518 has is another really important differentiating factor between PTC 518 and branaplam. So I think that there are, again, important differences in terms of the way the molecule is constructed, and that we really have a molecule that's specific and optimized for Huntington's disease. And I think, we just know from the fact that branaplam has been used in other diseases that we can't necessarily say the same. But I also want to point out that the other key difference is, while Vibrant HD is not moving forward, Pivot HD is. It's moving forward around the world, and we're going to continue to move that program along.
Thank you. And can I continue we have lots of great questions. If that's all right, I'm going to continue with some of them. So some folks have asked about mutant Huntington. I think there's one question about whether this was an allele-specific drug. Is this lowering total Huntington or mutant Huntington? And then maybe as part of that, you can also talk a little bit about the things that you are measuring as part of this trial. I think folks are wondering, about what are good clinical correlates? You know, is it mutant Huntington? Is it
Absolutely.
imaging? Um, and you know how that compares to what you saw in animal models.
Yeah, absolutely. So let me start with the first part of the question. So this is not allele specific. So it's lowering both the wild type and the mutant Huntington protein. And what we've done in this program is obviously come up with a target range of Huntington lowering overall 30 to 50%, which we believe will lower the mutant Huntington levels sufficiently to have an impact on the disease. And that's based on a lot of the literature and a lot of the research that's been done on Huntington lowering, as well as a lot of conversations we've had with Huntington's disease researchers and clinicians around the world. So we're very comfortable and confident in having an approach that is not allele specific. In terms of endpoints in Huntington's disease, well, I think that's a question that the community has been working on for decades. And what we're doing in Pivot HD is sort of taking it in steps. The first part of Pivot HD, those first 12 weeks, is really focused on understanding the drug levels and the lowering of Huntington mRNA and protein, the different drug levels between the brain and the blood, which is really important information we need as drug developers. It may not tell us right away the effect we're having on the disease, but it's fairly important for us to understand then what doses we can have that can ultimately have an effect on the disease. In the second part of the study, we start looking at what we call biomarkers of disease. Biomarkers are different measurements, whether those be of measurements of things in your blood, things that we see on X-rays, that start to tell us, are you affecting things that are associated with the disease? For example, we know that there's something called NFL, or neurofilament light chain, which is a marker of nerve injury and nerve inflammation. Over time, in people with Huntington's disease, those levels go up. Well, we're going to see if we can stabilize them, keep them from going up, or maybe even have them go down. That would tell us that we're starting to have a protective effect on the nerves that may get injured as a result of Huntington's disease progression. We're also looking at the level of mutant Huntington protein in the CSF, or cerebral spinal fluid. That's the fluid that bathes the brain. And the way that Huntington protein gets into that fluid is as cells are injured and start to die, they spill Huntington protein into that fluid. So we know that over time, people with Huntington's disease will have a gradual increase in the levels of the mutant Huntington protein in the cerebral spinal fluid. So we say, okay, if we're giving a drug that's slowing the injury of cells in the brain, slowing the progression of the disease, we'd like to see a lowering of those levels in this fluid around the brain, or the CSF. Because again, if the sign of progression is increase, then a sign of slowing progression would either be slowing increase, stopping the increase, or maybe even decreasing those levels. So those are the types of things we want to look at in the Pivot HD study. We also want to look at MRIs. It's well known that over time, that on MRIs, the size of different parts of the brain can go down, shrink, as more cells get injured over the course of Huntington's disease. So we want to look on MRIs, which are basically fancy X-rays, to see, are we stopping that reduction in brain size, which would tell us that we're, again, another sign that we're having an effect on the progression of the disease.
Thanks so much, Dr. Klein. And I think that we've touched on a lot of the main themes today, and I definitely want to, respect the time that we've set aside for this webinar. So, at this time, there are definitely some very specific questions in the chat that, folks can feel free to reach back out to HDSA, and we'll be in touch with PTC and our speaker today. So I just want to thank you one more time for joining us today. And I know Dr. Johnson would also like to say a few words.
Yeah, I just wanted to jump in and say, Matt, thank you, and to the PTC team for being able to come here today and to talk to everyone that's present, as well as the folks that'll be watching this in the days ahead. I think it's great to get an update from you all. And special thanks to Leora for leading this with you. I really appreciate the two of you putting this together and sharing this with the community. So thank you all. And thanks to all of you who attended it.
Yeah, thank you both very much. And obviously, please, Leora, send questions our way. We're happy to try to answer them. And incredibly grateful for the opportunity to share this update today, and look forward to being able to come back in the future and continue to keep the community updated on our progress at PTC 518.
Yes, we will welcome any progress updates. Thank you so much, Dr. Klein.
Thank you very much.